Morning. How are you doing? Good. Can't wait to holiday. Yes. Yes. Still get duit raya. I wonder during this time, like kids this day, how much you get for your duit raya? Ooh, 100. You know what? You should go next level now. Your aunties, your akong, your uncle want to give duit raya. I don't want. I want Bitcoin. Ask them to scan. Give now. Download lah e-wallet into your phone. Don't QR code do it now too. QR code for the apa ni digital coins. So okay, let's make a start. So we're still on the lesson of seed germination and dormancy. This is where we left off on Monday. Basically, the summary causing seed dormancy, including light, temperature, physical seed barrier, such as thickened seed coats, immature embryo. It's pretty much rudimentary, meaning that the embryo, the moment it gets detached from the mother plant, it is still developing. Remember the example of the chicken egg, right? When the mommy chicken, the hen lays the egg actually in it, there's no chick, but you got the egg yolk, right? But as the chicken incubate the egg, brooding for a while, then the egg develop into chicken inside of it, inside the shell, right? And finally, the presence of inhibitory chemicals of substance. And for your level, we are pretty much going to cover the hormone balance between ABA and GA. ABA is what? AB acid, abscisic acid, GA, granularic acid. Right, okay, that's good. Right, okay, so let's see. How can you break dormancy? Remember, okay, dormancy is a type. It's basically the seeds are put to sleep. Dormancy can take place in any organisms if they are able to do dormancy. The polar bear hibernate, that's a form of dormancy. In essence, dormancy is to significantly slow down the metabolic process and the use of energy at super minimum value, right? So the organism is still living, but the energy expenditure super minimum, and also the substance needed to grow and develop, they are pretty much inactivated. So that's the dormancy, okay, the resting period of it. If I'm not mistaken, if you want to look at the etymology of dormancy, it's what is it? I think a number of people in the past used dormancy in different form. Dormir, I think dormir, it means to sleep, and also French, yeah, to sleep, to rest, yeah, and then of course English changed it to dormant, right? So the seeds have been sleeping for quite some time. Then you want to awaken your seed. What do? Shake it violently. Yeah. Wakes up! It's Hari Raya. Go to mosque. How do you wake up your dormant friend? How? Right. Stomp on the face. Or you're very kind. Bangunlah. Is your friend going to wake up if you go that way? What's going to happen? Can continue sleeping further. Right. So to break the dormancy or to awaken the seed, it's not like waking you up from the sleep, but rather it involves a cascade of reactions, a chain of reactions. One thing leads to another, then you get your eventual event, which is the seeds awakening up in the form of spur in growth. But in order for this to happen, a number of things need to set in place first. And we're going to look at it. So, the physical of the seeds need to be overcome first because the activator is pretty much water, right? So the water is the signal and also the activator for various chemical in the seeds, okay? So the rehydration of the dormancies, which is pretty much dry by now, is achieved by the imbibition of water into the seed. And also the seeds, the physical of the seeds, as you have learned, there are many forms of seeds. Remember the double coconut, cocodimer? That's very big. How are you going to wake up the seeds? The embryo in there. You're going to knock on the coconut shell. Hello? How are you going to wake up the coconut embryo? What? Bang it against the floor. Actually, if you bang it, still it's not awakened. So, in many cases, actually, you need to do the deliberate injury to the seed coating or the testa, the seed jacket, so that there is a rush of oxygen into the seed as well as water. And then also it can be achieved if the seed in the soil with the presence of various microbes to decompose the seeds to a certain degree, and the water inhibition can take place, right? And when this happens, especially the rehydration process, this will cause the environmental effects to come in together. Before rehydration, the light can be present. The temperature, the correct light intensity can be present. That the temperature is optimum. The water can be there, but if there is no rehydration, meaning that the water manages to get into the seed, go to the embryo, optimum light and temperature is not going to do anything, right? So there's a combination of two things. Water gets into the seed, water inhibition, and then you get optimum and correct light and temperature. Then you're going to set off the germination chain of reactions. Then you're going to get the this is pretty much towards a bit more advanced in terms of awakening the seeds. When you get the hormones come into place, okay, and you're going to see why. Right. Upon seed germination, you're pretty much going to see the evidence visually by the emergence of radicle. Remember, radicle is the baby of which organ? Root. Right. Okay. So to break the dormancy, the seed need water, warm temperature, and if you do not want your seeds to grow just yet, you need to keep it dry and cold. And this is the very technique the seed bank used to keep seeds for very long time, 20, 50, 100 years. You know seed bank? We have seed bank as well, but it looks like tak tahu nak panggil cakap apa. Tak apalah. It's in the process to make it prettier. Seed bank, that's one in England, and that's another one. It's called Millennium Seed Bank in Svalbard, which just before the North Pole. Svalbard is an island. It belongs to the country Norway. right? So they got this seed bank, and this seed bank, they collect and receive seeds from various countries, and they put the seeds to sleep by doing this. They dry the seeds and then they make it cold. Okay, so what's the temperature like? Around minus 20. Yeah, minus 20, but it's dry, so it's not minus 20 like your freezer. Your freezer at home, that's minus 20 as well. Freezer, okay, not fridge. Fridge is where you keep your Vitagen. Freezer is where your mom keeps the fish, right? So this freezer at home is rather humid. You got your humidity and so on. But the minus 20 in the seed bank, it's dry. They control the humidity. Can be down to around 40% relative humidity, RH level. So the seeds can stay dormant there for quite some time, right? So that's a summary table of what's happening. Dormancy is caused by thick seed coat. This can be overcome by scarification. Example, scarification, action, nicking, digesting, scrubbing, fire. Remember the eagle story? Yeah, so the fire. Can you set the seeds on fire? Actually, in a way you can, but we don't really do it in tropical country. What example is that? Pine cone. Some pine people actually burn it. When they get the seed out of it, the seed actually ready to germinate. Right. Example, coffee tree. Yeah. Oh yeah, another action with the scarification. Freeze-thaw cycle. It's pretty much learned from nature actually. In countries that have distinct night and day temperature, for example, that the Saudi countries, in the Middle East country, during the day, it can be as hot as 40 degree. Yeah, during the night, it can be as cold as 10. So there's a big difference in temperature difference, right? So when this happens on regular basis, this is what happens in the freeze-thaw cycles. Thaw, in case you do not know, thaw means to melt. Okay, to unfreeze it, thawing, right? You have a frozen princess due to the bad wish. Then the pretty prince from the kingdom next door come to unfreeze the princess. That action is called thawing. Yeah, you actually, I think you can see your mom do that. They got the frozen chicken and then they soak in water in the sink. That's thawing, right? Okay. Do this multiple times. The substance going to expand, contract, it's gonna break, and then you have achieved your scarification. If the dormancy is the seed coat is not really a problem, so the light is enough actually to awaken it. For example, the lettuce, and temperature as well, dark, light and dark. Yeah. Light and dark cycle, we're going to see in a bit how this can cause the seed to germinate. So insufficient development, meaning that the embryo is still in the rudimentary form of it. Not only embryo, it can be other organelles in the seed. In the seed, there are mitochondria as well. And this mitochondria, the moment the seed is detached from the mother plant, it is there, but not quite fully developed. Yeah. The moment the seed is ready to do the germination, the mitochondria will mature and do the job. What's the job of mitochondria? Yeah. Generate energy. That's correct. Soil fungus association. This is a good example when I told the story about the orchid. Okay? Because orchids do not have one structure. What? That's the very reason why orchid seed is super small. It's lacking of what? Baru mention hari Isnin. Endosperm. Yeah, it hasn't got any food reserve. That's why it's super small. Right. Right, if dormancy caused by abscisic acid, you do the stratification and vernalization and most CT feral plants. For CT, don't say the CT scan of your brain. You see, when you learn science, you will see that sometimes certain reference material, they're not going to use Celsius. They use what? Fahrenheit. So you will see when especially if are you horticulture? Yeah, this is the reason I put this. You will see that many of your reference book is going to come from countries that don't really care about example in our country. Okay, so they're going to use the convention, abbreviation, naming based on their country. So CT actually refers to the a state in the United States. What's that? Connecticut. United States of America. You know USA? How many states they got? Cakap 40 tu dah salah lah tu. Dia tak ada 40 kat situ. So one of the many states in United States is called Connecticut. And the Connecticut is actually associated with a specific zone of temperature. And this is labeled as CT. Okay? Feral plants means, feral? Like a feral cat? Okay, that's the difference between the wild and the feral. Wild, from the beginning originally, from the jungle. Feral, the seeds actually maybe grow in domestic area, but somehow there's a surplus or excess of it, it got escaped from the garden. And then it grow uncontrollably. Now it becomes feral, like your feral cat. You take care of your cat, suddenly your cat elope and follow the boyfriend. Stay in the jungle. What was once your home cat now become the feral queen of the jungle. Yeah, when it comes home, do you think you can easily pet your cat? No. It's gonna eat you up. Yeah, especially your face. I've got scar in my hand actually. So that's why I quite remember the feral cat. If you come close to my hand, there's two scar here. One here. This is actually the canine of my feral cat. Then it bites. Lepas tu hanging macam tu. No, actually, I'm sure the cat is very sorry. He has been going on in the jungle for like almost a year. Suddenly came back home. Then I said lah, kau tak dapat makan kan? Lepas tu kau balik. And of course the cat ignores me and then continue to sleep and in the usual place. Then a few moments after that, when the food is ready, I tried to wake up the cat. Like the gentle petting. I think he forgets that he's in home now. He thought he was still in jungle, so it got spooked. Then the feral instinct comes in. Sian aku. Tak pasal tangan jadi biru. Yeah. The feral cat is actually they got, I don't know, some kind of venom or poison in the canine teeth. Your home cat is fine. Yeah. So feral, is it a problem? Yes, it can be invasive sometimes. And the inhibitor, like the phenolic compounds, this is leaching by the repeated rain. This example I have given as well. Okay. Like for example the cacti. Okay, let's see. This is the summary of the stages in the germination, which we are going to have a look at in details. Involves pre-germination, germination, and post-germination. We're going to look at these events individually. Right, so we started off at the pre-germination. Right. So upon the water imbibition, remember, okay, two events happen: rehydration And then the environmental factors, temperature, light, and so on, just nice for the seeds. So what's going to happen? The mitochondria are going to get reconstituted, meaning that the mitochondria was actually underdeveloped or immature. Okay, when the signal comes about, it's awakened, then the mitochondria will mature or reconstituted to do the job. Then what's going to happen? So in the endosperm, as well as in cotyledon, you got sugars, you got carbohydrate, you got protein, you got lipids. And this will cause all this food reserve to be used by mitochondria to kick off respiration. At first anaerobic, because there's less oxygen, then it moves with aerobic. When this happens, lots of ATP going to come about. When there's ample energy is present, RNA is activated. RNA is what? RNA, what? Ribonucleic acids. And why do you have RNA? What's the function of having RNA? Eventually, what do you get out of RNA? Protein lah, protein synthesis. And then you got your enzyme, then you got your further DNA synthesis. So you can see that that doesn't take very long. That's why when you germinate your bean sprout, your tauge, that can happen like, oh, that overnight can happen this much. This is what happened. So it's very rapid. Okay, and further mitosis, because the cells are now awakened and it's actively dividing. Okay, so that's when you see that your seeds now has swollen and the budding start to appear. Okay, either the radicle or the plumule. Right. What happened to the food reserve in the seeds? Okay, let's see. Oops. So the food reserve in the seed is pretty much inaccessible by the embryo. The food reserve is needed while the seed is growing, because at first the embryo is not able to photosynthesize. It is plant, but it is not able to photosynthesize. So the food reserve provided in the form of either endosperm or cotyledon will be the energy source and also the investment to be used by the embryo to further develop radicle, to further develop the plumule, until it is all used up and the seedling can be independent photosynthesizing on its own, right? So for this to happen, the mobilization of food reserve to happen, a set of enzymatic reaction needs to happen in sequence, and then you're going to see that. So we got starch. Starch is a complex molecule which is present in abundance in endosperm. Upon rehydration, seed imbibition, you're going to see that this is going to activate the alpha amylase. Alpha amylase is an enzyme that breaks down what? Carbohydrate lah. Yeah. So you get your maltose. Maltose, what kind of sugar is that? Yeah, so it's a disaccharide. Glucose and glucose. And that's a negative feedback mechanism here, meaning that the more maltose present in the seeds now, maltose is not there at first, but the alpha amylase breaks down the complex carbohydrate, then you get your maltose. This maltose will feed the negative feedback to the alpha amylase to become lesser and lesser as the maltose becomes more abundant. So once all the starch in amyloplast is hydrolyzed, the enzymes will stop to work, meaning that this alpha amylase is not going to do the job forever. Eventually it's going to stop because, number one, there is no more starch to be digested or hydrolyzed, and then the maltose has dominated the scene. Very strong signal for the negative feedback. Yeah. So you can see that the release of complex carbohydrate for the use by the embryo is determined by the demand, meaning that if you have germinating seeds in two temperature, the cool temperature and the warm temperature, you're going to see that the cool one is going to grow slowly. And if you measure the rate of alpha amylase and maltose formation, that is going to be a bit less compared to the warm germinating seedling. So of course the warm germinating seedling is going to use up all the food reserve first compared to the cool one. Okay, it's going to grow faster and more vigorous. Right. So what happens actually in the mobilization of food reserve? So the food reserve, there's so much in there. Okay, that not only the macromolecules like the carbohydrates, proteins, and lipids, you got vitamins as well. Vitamins, phenol acid compounds, and so on, antioxidants. Yeah, so much thing. Carbohydrate, you've got the amylopectin and amylose, like your rice. You know your rice? Rice is high in amylose and amylopectin. Depends on rice. The rice, the white rice that you eat in Malaysia, the one you can turn into biryani rice and so on, that is high in amylose. For your sushi rice, the amylose more or less. You get more amylopectin. That's why the sushi rice is sticky. Yeah, because the amylopectin content is high now. Yeah. So and then you got protein as well. Example protein is how do you pronounce that? Z-E-I-N.
Z.
Zinc. Zain or Zain. Either one. Zain. I'm British, so I pronounce it Zain. Zain. Tak percaya, tanya Google. Zain. It's a protein. So when it is hydrolyzed by a group of enzyme under the family of protease, you're going to get amino acids. While the lipids, such as oil, by the action of enzyme from the family of lipases, you're going to get fatty acids and glycerol. Okay, so everything is broken down so that the nutrients can be absorbed by the embryo and the embryo can grow happily, right? One important point to take here is that all this result of breaking down, it now turns into large insoluble macromolecules. Okay, it started off as a large insoluble macromolecules. Starch granules. The reason it is granule, because it is insoluble. Sugar, is it soluble or insoluble? Yeah. So this is the reason why the seeds needs to break down the carbohydrate so that it is soluble and it can get into the developing cells. If it is insoluble, that's not going to happen. Okay. Oops, sorry. Tak apalah, tengoklah. So let's see what happened. I hope this turned into so you got your seeds. Let's see the things that trigger factors for germination. So you've got light, the coolness, chilling, water, rain trigger, the activation of salicylic acid that is making the dormant seeds to germinate. You should know by now. There are a number of things that can happen to awaken your seeds, right? And see what happens next is you're going to see that the seeds, after taking up the water, the GA hormone is released from the embryo to signal alluron. Wait, one moment. Don't get too far. Alluron is actually alluron is a name of protein granules. Okay, so this protein granules surrounds the endosperm. Then the aileron respond by synthesizing and secreting the hydrolytic enzymes, like the alpha amylase, to hydrolyze the stored nutrients in the endosperm. Okay, it depends. Sometimes the aileron can release the lipase enzyme, okay, or the protease enzyme, and then the nutrient like sugar will be absorbed from the endosperm because it's in a simpler form now. It has been hydrolyzed by the scutellum or cotyledon, depending whether it's a dicot or monocot. And then the growth of seedlings starting to happen with the emergence of roots and then shoot. Okay, so then what happens? Is it the end of it? Not quite. Let's play the label. Don't look at your notes. Let's do the revision sekejap. So let's say that this is the ballistic. This outline, what do you call? Seed coat. This is the seed jacket. Yeah, fruit or the seed coat. Then what is this? Endosperm. Yeah. What is this? Yeah, embryo, cotyledon, the whole of it. It can't be cotyledon because it's a monocot. What do you call it? Cotyledon in the monocot. What do you call it? Scutellum. The shield. Shield too. Ni, shield ni. Scutellum. So that's the embryo. Okay. And what is that? Oh, that's the shoot apex. That's the radical apex. And that's the cotyledon or scutellum. Yeah. I mentioned on Monday, right? If you call it cotyledon, it's correct as well. But if you want to get it precisely, it's called scutellum. Right. So the water will get into, okay, tell me the water will enter through what? What structure? The entrance that is equivalent to your belly button. What was it called? Line apa? What? It starts with M. M for Malaysia. What? Micropyle. Janganlah lupa. Micropyle. Okay, then water imbibition will happen. This is when you get your rehydration here. If you refer your notes on the earlier slides, ada slide yang label micropyle. Okay. I don't want to go back sebab ni ada animation nanti lambat nak ke depan. Lepas tu tak ada pula note aku nak tunjuk kat sini. Okay, so you have your complex carbohydrate in the form of starch in your endosperm. Then what will happen? You will get hydrolysis. What's the product? Sugar, maltose. Then, okay, now what is this thing? This thing. In the form of grains surrounding the endosperm. What's that? I just mentioned 3 slides ago. This is the aileron. Okay? Aileron layer. Aileron is actually protein grains. Protein, motor. So collectively, it's called aileron layer. Okay, so aileron, storage protein, it breaks down. What do you get? Amino acids. This is your basic biochemistry. Okay, tak boleh lupa. Kena tahu. Complex molecule. When it breaks down, you will get the monomer or the blocking. Not the blocking block. The building blocks of it. And the building blocks for a protein is amino acid. Now you get your DNA. DNA in the cells of the seed will do further transcription, and then you get your RNA, and you will get translation to have more protein that is called alpha amylase. Alpha amylase breaks down who? Starch. Right. And this alpha amylase, after it has breaks down the complex sugar, you get your maltose, right? The sugar. Your sugar will undergo exocytosis, not exorcist. Exorcist, what you call your padre when your sister next door screaming non-stop. You know exorcist? Apa ah? Itu je ke dia tahu? Apa exorcist? Kau dia tak tahu exorcist tu apa? Bulan puasa tak jadi kot. Right. Bukan exorcist, exocytosis, meaning that the substance fuse to exit out of the compartment in the form of vesicle. Then it gets released out. Right. It nak release ke mana? Pergi ke embryo. Yeah. Then embryo will start to eat that up. Then it will start to grow, and more GA will further get activated, because when GA is abundant, ADA is suppressed. And remember, GA is needed to spur the growth. Right. Capsella seeds. Capsella, I do not know whether you got the plant here. This is from the mustard family, Brassicaceae. You know mustard? Mustard plant? Yeah. This is Capsella, the cousin for mustard. In case you want to see the micropyle, ni. Like the belly button for your seed. So let's see. What structure is that? Seed coat. Lepas tu, aku cakap lupa saja. Lagi? Apa ni? Endosperm, right in the middle. And embryo, let's see. What is this structure called? Apical apex, and these two, ada dua. Cotyledon. So this is monocot or dicot? Dicot. Right. So, and this is hypocotyl. Why is it called hypocotyl? Below cotyledons. Below cotyledon. Hypo means down or below. Epi means up or on the edge. Kan? Meso means middle. Ingat tu. Right. And this is your radical. And dekat hujung radical, you're going to get what? Root apex. So, we've got two apices here. Shoot apex and root apex. Okay? And both are meristematic, meaning that they can give to further growth. Right? Let's see lettuce, because just now you have looked at the barley, right? That's monocot. What's this? Sikut. Tekan, tak apalah. What's this? Okay, iyalah, ada dua tu. Semua ni apa? Apa monocot? Lettuce. Dicot kan ada dua tu. Radical, apex, cotyledons. Sebab tu nama dia dicot, ada dua cotyledons. Shoot apex. So it's a junction here. Sebenarnya benda ni, I'll redraw for you. Dia kind of like baby fetus in mommy's womb. Kind of terpeleot. Dia tak straight. Macam, you know, running kids. It should look like this. Ha, macam tu. Ni. It has been folded because the seed, is it a big space?
No.
No. Look at the fetus. The womb, is it big? So the baby, is it lying down in the womb? Kena folded?
Folded.
Macam mana tahu folded?
Sebab tempat dia tak besar.
Sebab tempat tak besar. Lepas tu, kau dah duduk dalam tu lama-lama. So it's folded because of the constraint in space. So this here is actually this. Okay? And this is what you're going to get your plant seedling eventually. Right. Okay, water imbibition. Then you're going to get pretty much like what you saw in Bali earlier. DNA transcription to give rise to RNA to make what? Alpha amylase. And alpha amylase will do what? Sugar to do the from starch. That's correct. Hydrolyze, you get your sugar. And then that's when you get your growth of your radical. This is why the moment the seed germinates, you're going to see the radical first. This is the reason. Right. Okay, we'll have a look at this about the phytochrome. Phytochrome, PRPFR. Nak ajar? They have learned. Tak tahu dia ingat ke tak? Okay, let's look at the phytochrome. Yeah. In plant cells, on the surface of the cell wall, floating about in the cytoplasm, residing on the surface of the organelles such as nucleus, it can be the presence of so many different substances. And usually they are protein or glycoprotein, meaning that you got the starch and protein comes together. It's called like glycoprotein. Okay, and all of these things, they are actually not doing anything except to receive signal. To receive signal to activate itself so that it can trigger the process of other things. So you can think of it like a astro antenna. Receive, send signal so that your mom can watch astro. Pretty much like that. So phytochrome, it's from two words actually. Phytochrome. Phyto means plant. Chrome means color. So phytochrome is actually a type of protein residing in cytoplasm or nucleus, depending. Okay, it is not static. Important for you to understand this. So we call this pigment proteins. It's got color, it absorbs color, and it does colorful things. Okay, and there are many pigment proteins in plants. All right, chlorophyll. You know chlorophyll?
Yeah.
What color is that? Green. Chlorophyll never alone the color itself. Chlorophyll is actually present in the form of protein complex. You got the color part and then it's attached to protein complexes surrounding it. Right. So, what's the function of phytochrome? Let's see. Salah. Phytochrome is got two form of it. P stands for phytochrome, got R, then you got phytochrome FR. Tengok ni ya. Please pay attention to this but even until Viva PhD, people still not getting this. Alright? This R is actually referring to different shades of red. Okay? You know your rainbow? Rainbow got color, right?
Yes.
What's the color of rainbow?
Red, orange, yellow.
Huh?
Yellow.
Yellow.
Blue, purple.
the either one lah. Okay, so this color is the qualitative assessment based on your eyes. You see it. Other way to look at it, you can actually have the quantitative association to this color, meaning that you can get the nanometer of the wavelength that enables you to see this color. For example, we start with blue here. This is actually 380 nanometer. Nanometer is referring to the wavelength or lambda. And then all the way to red here, you can get up to 700 nanometer. So from 380, or for simplicity, I'll just put 400 nanometer. With the increment of nanometer value, you are going to get the shift of color from blue to green, yellow, orange, and then to red. Also to highlight, from 400 to 700, even though it increases in value, absolute value number, the energy becoming less. So it's opposite. Lambda, nanometer, sorry, I'll just nanometer. When you have high lambda, you have low energy. When you have low lambda, like here, you have high energy. So for your level, let's take that first. Okay. This is why you cannot look at the sky for too long. What color is the sky? Blue. So based on this, high or low energy? High energy. But the same sky, look at it 7 o'clock. What color is that? Red, orange, right? Can you look at the sky for longer now? The reason you can look for longer, because the wavelength is higher, 700, and the energy is less. So it is less damaging to your eyes. That's why you can enjoy sunset. Oh, right. Okay, I don't want to talk too much about the whole spectrum of color. I just want to highlight about this red thing. So the color red start from about 740 until only God's know whatever nanometer. Okay, so as you go to this side, it gets deeper red. And for our human retina, you got retina in your eyes, you can actually see up to 700 nanometer. Okay, lighter red. This is red. The red here is about 660 nanometer. So it's just around here. 660 nanometer. Far red means that whatever value of red color beyond 700 nanometer, far red. So in this case, the far red is 730. Somewhere here, and go, you got 730 nanometer. Right. So why this is important? The same protein, the phytochrome, it has two forms, active form and inactive form. In the inactive form, it is in this side, inactive. And in active form, we call it PFR. So how can this guy get inactivated? When it is inactive, we know that this protein is in darkness. But suddenly, when the sun comes about or you shine your red LED to it, this phytochrome protein will receive red that you can see. Now it gets activated. The moment it gets activated, it is now called PFR, the activated form of phytochrome, right? If you make it stay longer or suddenly the red lights is off, the activated phytochrome can revert back to the inactive form of it. And the inactive form is called PR, phytochrome red. So very simple thing to remember here. Phytochrome is a pigment protein. It has inactive form and active form. You get inactive form in darkness. The moment the light comes about, which red is present in abundance, PR inactive is converted to PFR, which is active form. When this has been going on for quite some time, or suddenly when the light is off, PFR can revert back to PR form. Okay, so 730 nanometer, you cannot see. When you cannot see, it is bright or dark? Darkness. So meaning that 730 is the red that you can't see. You cannot see, but your camera can see. Right? Okay, why this is important? Phytochrome is a protein that can trigger seed germination reaction. It signals GA and many other things in the cell to awaken. Because now I have been activated, you guys need to awaken as well so that these seedlings can grow the radical, can grow the plumule, make the shoot and do the rest of it. Right. Okay. So this is why it is important. And mind you, not all plants do this. The lettuce that you saw earlier, it uses this. Get it wrong. Yeah. And this whole thing, it's called photoactivation. Means that the germination of your plant is not going to happen even if you have provided with water, right temperature, nutrient and so on, until the correct red light comes about. And when this happens, photoactivation has taken place, then the rest of it will follow suit. Right, okay. So I just put it this to repeat back what I have explained to you. Again, okay. I repeat again. So this is pigment protein that has been extracted. It's actually in the cell, in the cytoplasm or nucleus. But scientists extracted it, put it in a vial so that you can see. What's the color? It's got two, cyan or blue-green on the PFR side. So when it's inactive, the PR here, it's actually bluish, the solution, the pigment protein, when you extract and put it in a vial. You shine the red light upon it, then suddenly the vial change color. When it changes color, it means the phytochrome has been activated. The color evidence of it, visual evidence of it, it's now becoming, what color is this? Gray. Cyan, turquoise, something like that. It's a bit on the greenish side of it. If the red light is going on for quite some time, this PFR is going to get kind of like too much of a good thing. It's going to break down. Okay. However, if the red light only happens for quite some time and then it's off or it goes away, you will start to get another kind of red in darkness, which is 730 and beyond nanometer. When that happens, the PFR, which is the greenish solution, will change back to the bluish solution. So it is still phytochrome, but in inactive form now. So what will happen? This is the response that you're looking for. When phytochrome is active, you're going to get this. You're going to get seed germination, stomatal opening, as well as the control of plant flowering. This is why sakura knows when to flower, to bloom. You know sakura? Malaysia, Malaysia ada sakura ke? In Malaysia, it's called tebubuya tree. You know? Tebubuya. The rose tree. Sakura, it blooms in spring, right? Before spring, what is the season? Winter. What's the situation in winter? Is it bright? It's dark and cold. That's winter. Otherwise, Santa wouldn't come.
The Santa exists.
You look at all the Christmas movie. Is it super bright when the Santa comes? Santa comes at night. During winter, you only have a fraction of daylight. Most of the day is dominated by night time. Yeah. For example, like we are fasting now, right? If you go to England now actually not that bad during summer when during my student time. I start fasting suhoor at 2:30 in the morning, and iftar break the fast 9:30 p.m. So you tell me how much hour left for me to nak semayang maghrib, nak tarawih, nak sahur balik. That's like only 5 hours. Yeah, memang tadi tu. Yeah. I changed my working hours during Ramadan when during that time. Masuk kerja pukul 12 malam. Actually after tarawih usually ends 12:15. Dan balik kerja pukul 10 pagi. Orang masuk kerja, aku balik kerja kan. So that's summer. Winter is the opposite of it. Yeah, in winter, subuh starts. That's quite normal. 6:40 fajar, but you'll get your maghrib 3:20. Maghrib ya, 3:20. 3:20 dah maghrib, dah malam. And then isya pukul 5. The rest of it night malam. Right. So that is causing most of the phytochrome in plants in PR form, phytochrome inactive form. As the day progressing, you are approaching spring, the photoperiod gets longer. And photoperiod gets longer, you will get more of this or more of this. Blue or more of green? You're going to get the green, the active one. When that happens, that sakura will start to be activated, awakened, and then it has enough energy. Why it has enough energy? Because PFR control the flowering. Yeah. Then it will start to bloom. It maybe it's unusual to you because the sakura plant, the cherry blossom plant, or let me call very well, Inghua. Yeah. Is it right? Inghua? Ke dia tipu aku? Inghua lah, bunga cherry. Hua tu bunga, ing tu cherry. Ni chưa tao. Okay, so sakura has pretty much foliarless, gondol, tak ada daun. Suddenly when it comes back to life, leaf is not coming out first, flower first. So that's a special thing that is not happening in our country in tropical. So this special phenomenon in sakura, flower first, leaf later. That's a terminology for it. Is called. Oops, tak ada. Why I keep on getting marker yang tak ada life to it? Hysteranthus. Nanti look it up lah. Tengok apa benda hysteranthus tu. Uh, uh, when I teach botany, I taught this. Right? Okay? So, is phytochrome important now? It's very important. Do you have phytochrome? Ada tak?
Every flower appears before the leaf.
Apa benda kau mimpi seseorang? Apa? Dia tak percaya aku cakap. Kau yang aku scam.
The flower appearing before the leaf.
Betul lah aku cakap Sakura. What comes first? Flower or leaf first? Mesti betul kan? Hai. Tak apalah, he wants to validate me. That's what you need to do in life. Kan? Ini orang bagi sikit link kau klik. Lepas tu pelik duit tinggal RM1. Don't do that. Don't do that. Don't be so gullible. You know gullible? Ini dah jadi kelas English dah. Do not be gullible. Tengok. I highly encourage semua orang untuk check individually apa maksud gullible so you do not become this. If you are gullible, you are the one who only have RM1 left in the account. So are you gullible? Sometimes. Kan? Puji-puji sikit semua bagi. Janganlah jadi gullible. You are becoming scientist. Scientist, not only that tak boleh gullible, you cannot be naive. You need to be inquisitive. You need to have an ethical while working. So these are all the characteristics of a scientist, right? So develop, inculcate, nurture these qualities from the beginning. And I think when you graduate, you are above everybody else. As simple as that, right? Okay, so let's see. That's the first summary. So germination of seeds. So this is pretty much the summary of it. So you got the utilization of stored reserve. Remember, starch jadi sugar, lipids can become sugar as well, protein becoming amino acid. This thing, lipids, fats turn to sugar. This is actually not straightforward. The lipids will be broken down into what you saw just now by lipase. When lipid is broken down, what is the product? Fatty acid and glycerol. This glycerol can undergo another pathway to become sugar. That's how you get sugar. You are fasting, all right? Right. A month ago, you got 2 kilogram of fat kat sini. No, dah tak ada. Pergi mana? Oh, memang tak ada since the beginning. Your fat has been used sebab your body breaks it down to get the glycerol, and then that glycerol can dapat jadi gula, and gula can be used for your cellular energy, right? Okay. And germination of seed, transport a compound into the seedling. Okay, these compounds have to function, support respiration in the embryo, and also to provide the building blocks. The embryo needs energy. Okay, in order for it to need energy, it needs to do respiration. So that's why it needs ATP, phosphate, precursor and so on. And also it needs the building block, C, carbon, nitrogen for the seedling. The plumule, the leaf, the radicle, they are pretty much, I call CHO backbone. You know CHO? Not CHO, your friend. Carbon, hydrogen, oxygen. Pretty much CHO. Right. And you also need it for the expansion and growth of the seedling. Radicle elongates down, hypocotyl expands up. Then you got the root system and emergence of the shoot. Roots is very important because in you, that's your intestine and your villi. You ingest your food is digested. The very structure that absorb the nutrients from the food is in your intestine, the villi. So this is why the plants invest so much in developing proper radical and roots network, because this is where they absorb the most of the nutrients that coming. Okay, so I do expect you to be able to draw something like this. You need to be able to draw now. I know masa lab nak kena draw, tapi draw lah kan tak ada apa. So you are able to draw something like this, and then not only drawing, labeling. So suddenly kalau dalam exam nanti, by the way, final exam is not from me, okay? It's centralized. So I cannot really help you. Yeah. Why? Tanya Marani. It's centralized lah. You got other groups. Final, I cannot help you. Just one test tu boleh lah. Right. So suddenly you are being presented by this, and then you are asked to label. You need to be able to label. What is hypocotyl? Kalau hypocotyl kat sini, cotyledon kat mana? Mestilah sini. Sebab hypocotyl down. Then you got your root. Hypocotyl. Where's the cotyledon? Up. That's why it's called hypocotyl. If this is hypocotyl, this is what? Epicotyl. Epi atas lah, sebab cotyledon kat sini. And then you got roots, primary, and also the secondary roots. Okay. And finally, the type of germination. Let's see. So this is pretty much what I have said. So this is something for you to read as your review. How can you quantify the seedling establishment? A number of ways. The length, fresh weight, and dry weight. We are going to do this during your lab. Anyway, okay? But read so that you get some idea what's going to happen during your lab time soon. Types of germination. You got two. Epigel, hypogel. Epi means above. Gel is actually from the word geo. You know geography? Geography is study of what? Land. Land aja. Earth, the earth itself, geography. So when it says epigel, above earth. Who is above earth? Cotyledon. The other one is hypogel. Hypo means below or down. Hypogel means who's under the ground? Cotyledon. Still cotyledon. All this thing is referring to the positioning of the cotyledon once the seedling has germinated. Ni, benda ni. Ni.
So the hypogel is the monocot lah.
Yeah. Type of germination. What is this? Epigel or hypogel? Look at the cotyledon on the further right. Epigel. Why? Cotyledon up or down? So dia epigel. What about this? Why? Yes, cotyledon dekat bawah. Jadilah hypogel, bawah bumi. Right. Okay. Ini obviously aku ambil dekat planet pula. Dekat Google saja nak mencarikan suasana. Bacalah. It's pretty much what I have said so far. Right. Okay. I think that's pretty much it. That's the conclusion for you to read. Yay, habis. Bukan habis semester lagi. Nak test one? Apa? Tiba-tiba Google Form. Okay, right. Any question? What time now? 9:43. Okay. What's the date today? I got a number of things that's going to happen today. Sekejap. Pak Haji come when? 11. Are they going to visit the ladang as well? Ke dia nak visit the equipment? Alright. Okay. I got my own ladang. My ladang nothing moving or clucking. It's silent and peaceful. Crop science, okay? Not animal science. Dia tak ada bunyi. Okay. Any question from this lesson? No? Okay. We got two choice. Next lesson would be about water. I think I have inserted dalam your ni. Water. Oh, ada ni. Nanti tuan print ke? Tulisan dalam tu dah. I give you choice. Do you want to stop here so that you can revise and bertenang and be happy with your life, or you want to learn the water and then learn the water next Monday, or you want to learn the water today and be miserable?
Half water today.
Miserable first. Actually, if I teach, cannot stop.
Is it that hard?
Entah, aku okay, aku faham je. It's actually nak ajar water potential. Yeah. Tadi kan you or you want to get introduced first? Okay, let's go half an hour. Okay, tapi I have not printed this. Tak apalah, nanti bagi hari Isnin ya. Can you get this lesson? Masuk. Sekejap. Take 5, you can go to toilet apa benda semua, jangan minum. Greek alphabets. The one that you're familiar with, your ABC 26 letters tu, itu Roman alphabets. Roman alphabet ke? I don't know, making a story now. So Greek alphabets, pretty much like your regular alphabets, it has its own letters. Salah. It has its own letters, and also the uppercase capital letter and lowercase form of it. For example, the alpha, this is the capital letter, and this is the form of the lowercase. You got alpha, beta, gamma, delta, epsilon, and all the way until omega. The end of it is not Z. No, because this is Greek letter. Why this is important? You are going to be a functional scientist. In the science world, label is used regularly. Use Greek letters. Regardless in medical world, in the pure science world, chemistry, physics, semua ni gunakan these Greek alphabets. So if you're not familiar with it, it's kind of hampering your understanding nanti. And you need to understand some letters are universal label for a variable. Okay? For example, lambda just now. This lambda, is this lowercase or uppercase?
Lowercase.
So what's the uppercase? How does it look? Which one is lambda? Yang mana? B terbalik. Ni B. Line berapa? 1, 2, 3, 4, 5. 3. Ni. So ini lowercase. So dia bukan macam ni. Uppercase. Ini bukan nombor 8. If you learn Arabic, that's number 8. Bukan nombor 8. Or bukan inverted V. No, that's capital case lambda. So in physics, you have learned a number of variables yang ada this kind of letter symbol. Okay, you got lambda, what else? Rho, theta you learn it in trigonometry to represent degree of any angle. Apa lagi? Pi. Yes, pi. Yes, omega. What about delta? In mathematics, delta means what? Apa degree pula? Remember you learn your differentiation and calculus? Ini patut ada. To show difference. When one value minus one value, T1, T2 minus T1, equals to delta T. Maksudnya the difference between 2 points of time. Okay, so all of that has symbol, including now water potential. So which one is the symbol of water potential? Psi. You see, you are about to sigh a lot while learning this. The name gives you the hint. You're going to sighing a lot. Sigh. So, macam mana nak tulis? Salah lagi. Sigh. So it looks like this. Ataupun boleh tulis macam ni. So the lowercase and uppercase looks pretty much the same. So that's good for you. The sign. So the question remains, why use psi as a symbol for water potential in physiology? Why? This actually from an object. Trident. So trident. Who uses trident? Aku. Aquaman. Scientists dulu they like to use Greek deities and goddesses as their inspiration. So since we are talking about water, it's only sensible for them to use the gods related to water and ocean, whose name is Poseidon. Poseidon. So dipendekkan jadi Psi. So the power of the ocean. Boom. Poseidon. So bentuk Poseidon, if you read the story, dia ada weapon dia. Apa weapon dia? Trident. So they change it into Psi. Cerita dia. The English version of the name of the planets. Jupiter, Saturn, semua tu, itu kan nama Greek god semua, kan? What about ni Venus? What's that in Malay? Planet Venus. Zuhal Saturn, bukan Satan. Aku nak sebut salah dah. Saturn, bukan Satan. Saturn. Zuhal lah. Venus planet apa? Zuhra. So Venus is the goddess of what? Is the god of love. Yeah. Tak jadi check lah. Kau suka ingat aku scam. Tengoklah, Venus is the god of what? Venus. F. So your friend found it?
Roman goddess of
Love and beauty is effort.
Bukan. Jangan salah Tuhan. Roman. Oh, bukan Greek. Roman. Okay, fine, Roman. Okay. So if you go global later, you study abroad and so on, you do not get culture shock with this kind of nomenclature system. Okay, they are very fond of using all these Greek mythologies, Roman figures and so on dekat dalam science, in the way they are naming things, that kind of thing. Okay, right? So next time I want tanya, psi is due to what? Don't say Aquaman. That's fictional, right? Okay, psi. So let's have a look at water now. Right. Okay. Learn about water. Shui. Or Yao Ping Shui. Ada tak? Ping. Ping Shui lah. Air sejuk. Bukan Feng Shui. Okay, water. Do you like water? Mesti tanya. It's the question of whether you like or you need or you want or you love. No, I like to take a time explain water because this is an input in agriculture. And if you do it correctly, you can do so much with water at the farm level and also when you are working in the laboratory. Just by understanding the water, the properties of water to make it function. I do microscopy work as well. So my microscopy images always look very crisp. Sometimes I just play around with the water properties. Okay, not to tune in the microscope itself, right? So that's a good virtue that can come up by understanding the water, right? So water, H2O. Where does it come from?
One hydrogen.
No, originally, this Earth, before Earth first created, what was it like? Asteroid floating about. When Earth was first formed and created, it was just a rock, hot molten rock. Okay, there's no life to it. Okay, how on earth we have so much water covering like 2/3 of the planet? Where did it come from? Daripada mana datang? Underground. Okay. Before underground, where it came from? Can you create water? If on Earth you cannot create water, where water on Earth came from? Do you understand my point? Yeah. Where? Asteroid. Unfortunately, we do not have that Doraemon time machine, right? Otherwise, we can go rombongan kelas untuk bagi tengok what happened 4 billion years ago. Potentially, there are two explanations for it. During the early formation of the Earth, there is a violent bombardment of extraterrestrial objects from the open space, including icy comets. Comets, meteorite, and boom, come to Earth. And this comet is ice, actually. Ice. Okay, so ice, boom, melts because the Earth is hot. Then you get your lake, Tasik Sedang. Got it, one. And another is because of the early formation of the Earth, super hot. That hot is actually energy. And this energy somehow is enough to fuse oxygen and hydrogen together.
Oh.
Yeah.
But during that time, oxygen still present at Earth.
Oxygen can come about from the element before it. Look at your periodic table. You start off with what's the first element?
Hydrogen.
two hydrogen come together, what do you get? Hydrogen 2. Tapi kalau hydrogen tu one electron, one proton. What if I increase the atomic number of it? What do you get now? Nah, tak ingat periodic table. Helium. You suddenly get a new element just by increasing the atomic number, and then this atomic number can be added because of energy, because of the compression, so many things that can happen. You started off with one element, now you get a new element. So oxygen not necessarily have to be created from the flick of a hand. It can come from a free precursor element. Okay, so you add more electrons to the element, the valence of the electron will start to increase. You got two electrons in one valence at the beginning. We call it the duplet, and then you got your octet, and then you get some more, right? Eventually, you got your oxygen, hydrogen, and so many other things, right? Right. So, with this water come on and about, it has got properties like any other things, like the wood here. Does wood has properties?
Yes.
What properties wood has? Hard. Lagi?
Erection.
Apa? Apa? Flammable. 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 That's actually for chemical compound. Burnable may be right. What else? Properties of wood. So it can act as an insulator. Right. What about water? So these are the common properties of water. I'm not saying this is an exhaustive list of water properties. There are many more. But these are the properties of water. Let's see. So water got hydrogen bonding, means that water can become a universal solvent. Because in the water molecule, you got the electronegative part and then a bit more positive part. So this can somehow orient itself to get attracted to other molecules. And when this happens, there's a fusion of molecules, and the molecule which was once a solid can now dissolve and be part of the liquid. Easy example, sodium chloride. Right. And the water also have a thermal conductivity. Means that if you have like a container of water, if you provide heat from one end, the heat can travel from this end to the other without you having to stirring the water. Okay? Kind of like when you first learn about the sound. Remember your teacher tell you your sound can travel in solid, air, and liquid. Which sound travel the best? Solid. Can you listen? But if you put your hand kat sini, you can hear it. Right. So the water in liquid, if you put a sound wave in it, it can propagate and the sound can reach the other end of it. Right? And the same goes to the heat. The heat can travel. No need to do the stirring. And then the high heat capacity. H2O is not easy to break the bond. Means that before the bonds can be broken, a large sum of energy needs to be absorbed first, right? Meaning that the phases of water from solid to liquid to vapor or steam gas, you need lots of energy. That's why when you turn on your stove, cooking stove, Does your water in the kettle get boils under 10 seconds? It takes some time, right? Yeah. This is because, number 3, it has a high heat capacity. It is able to absorb so much energy before something happens to it. Right? And this is why your body is filled with water. And your temperature can be regulated easily by your body. Your temperature, what's your body temperature? 15. Are you fish? 37. Yeah, because he's Aquaman, right? That's why he's 15 degrees Celsius body temperature. 37. It's easy to regulate because we are highly filled dengan air. And the same thing happened to our planet as well. Like over 2/3 of our planet is water. If this is not the case, we are pretty much going to turn into a fireball. Because there is no big body to regulate our temperature. And this is the reason when I mentioned the plants transpire, it can cool itself. Number 1, it's not easy to increase the temperature of the plant because plant is filled with water just like human. And secondly, the moment plants transpire, the water that leaves the plant surface will pull along with it a large amount of heat from the plant, bring to the atmosphere. Right. So, perfect for cooling purpose. Cohesion. So cohesion actually should be number 4 and number 6. It kind of comes together. Cohesion and adhesion. Have you learned this before?
Yeah.
What's that?
I remember.
Can't remember. Cohesion is the sticking of similar molecule together. Hydrogen, water, wood to wood, similar molecule. Addition, the sticking of different material or substance come in contact together. For example, water molecule and then the xylem wall stick to it, right? So when this is translated into the water transport in plant, cohesion causing the water to be pulled from the bottom all the way to the top, right? And it is also not easy for the water to come back because it is sticking to the wall of the xylem due to the force of adhesion. Yeah. An easy way to remember is cohesion is cooperation. It's easy to cooperate among your same kind. Water, it's easy to operate. Water xylem, well, they need to agree first before they can do cooperation. Right? Okay, what else? Surface tension. This thing. What's surface tension? Ketenangan, ketegangan, ketegangan keadaan, permukaan. Give me one example of surface tension in nature. In nature pun boleh juga. Because you are patriotic. What? Droplets on the leaf. Okay. Lagi? Insects walking on the pond surface. Why?
Because that's high surface tension.
Surface tension is actually the ability, it doesn't only apply to water, to any liquid. Okay? So it can be to ethanol, it can be to sugar, it can be to your air vitagen. The ability for you to spread the solution, the liquid, before it breaks. Right? Since water have this hydrogen bonding, it is not easy to break the surface of it. So, water in the pond forms a form of skin on the surface that enables the insect to stride upon it. Hence the name of the insect, water strider. What's that in Malay? Water strider. You know water strider? Have you learned your entomology? Soon you're going to learn about that insect. Entomology. Water strider. I don't know in Malay apa. I do got the video somewhere. Water strider. Okay. Heat of fusion. Have you learned this before? Heat of fusion. Didn't you take your physics and biology before? Okay, chemistry. You during your form 6 ke, matrix ke? Lepas tu belajar tak? Okay, belajar. So what is it actually?
Changes of energy.
So the energy needed to change the substance from solid to liquid. From solid to liquid maksudnya from ice to water without changing the temperature. So we call it the heat of fusion. The other one is heat of vaporization. The energy needed used to change from liquid to vapor without the change of temperature. Okay. Right. What else? Polarity and solvent. This is actually I've explained in a way. Polarity because of H plus oxygen 2. That's electronegative in the water molecule. And then solvent. Because of this polarity, water is able to become the solvent. Can water dissolve sugar?
Yes.
Yes. Pretty much, if you think about it, water can dissolve any powder. Right. Yeah. Except gold powder. Well, gold is an element. It's not really powder. It has been powderized. Right. Okay. So I'm not going to go down there. Nanti kau marah aku. All this dielectric constant. This is actually for the other class, advanced physio. Yeah. Right. What's that?
Polarity.
Water. What letter is this?
Delta.
Okay, delta. Why delta? Uppercase, lowercase. So, is it the delta the difference? Partial. It's a symbol of partial. Partial. Okay. Partial positive charge with the hydrogen and then partial negative charge on this side. And then you got an angle to it. So electrons are pulled towards the oxygen. So this is why hydrogen bonding can happen. Water molecule stick to other water molecules, and then water molecule can stick to other molecule like the sodium chloride. Okay, this is the reason. So regardless of whatever you put, water get it covered because it's got both charges. Right. So as a revision, covalent versus hydrogen bond. So hydrogen bond, this is the hydrogen bond between the hydrogen and something else. What about the covalent bond?
Between hydrogen and oxygen.
Yeah, so that's actually the one that ties between the non-alike element in this example, between the oxygen and hydrogen. Without giving any table to refer, if I were to ask you, which energy you need more to break? Covalent or hydrogen?
Covalent.
Why?
Because the molecular forces is stronger.
Dah macam bunyi Google. Tapi Google versi Muslimah. Baguslah, at least dia tak tidur. Okay, right. Yeah. So this is to give you the perspective about the energy and the relation to the bonds to be broken. For example, like this. Okay. So you got your ionic bond, covalent triple bonds, all the way dipole interaction and hydrogen bond. So the bond energy, okay, bond energy means how much energy is needed to break the bond. Okay, you can see that ionic bond compared to hydrogen bond is rather weak. Okay, it's very easy to break it. So what is the reason? Why suddenly you can see that the covalent triple bonds, the value is so higher compared to hydrogen bond? Something to do with the molecular arrangement. You can see that CC, there's only one here, so you need less energy. When you got the double bond, you need to deal more per the same area, per the same number of molecule. And how do you quantify this? Joule per mole. Okay, or joule. I'm not going to go into much of this. Just enough to tell you, joule can be expressed in many ways. One of it is joule per mole to tell about the bond energy. And the joule itself, this is how you get it. Kilogram times the square meter over second squared. Right? So it is actually equal to the work done by force of 1 Newton acting through 1 meter. So that's very complicated, right? A joule. Where else do you find joule?
In food.
In food. What does it tell you?
Energy.
Energy. How can you, where do you think in agriculture you can use joule? Where? Apa? Electricity, penggunaan elektrik. When you look at the sun, that's energy, right? Can that be quantified in terms of joule? So that's actually called the solar conversion. You can use joule or watt to quantify the sun radiation that coming to the earth. Let's say that comes to the earth like 9 megawatt or 90 megajoules. For that area, you harvest your potato and then you only get 10% of it. 0.9 megajoule or 9 joule. 9 out of 10%, 9 joules. So what does it tell you? The solar conversion of your crop while receiving 90 megajoule is only 10%. It tells you there's a room of improvement. Yeah. So this is why agronomic practice is in actively researched and studied, because we are always trying to understand how to increase the solar conversion. People do breeding, people do fertilizer manipulation, all in the effort. The same piece of land, you cannot suddenly increase it, but you increase the number of solar conversion. Right. Yeah. So, how can you increase the solar conversion of your crop? Potentially. How do you think? Shona? Simple. Just by increasing the photosynthesis. The more photosynthesis you have, the more sugar you will end up. The more sugar end up means that sugar isn't sugar base. Glucose. CHO. So the more CHO that is present in your sugar, the more that you have captured and convert the solar energy into harvested organs. Okay, so there are many ways to do it, and that's something that you can learn later. Right. Okay. Specific heat. So it is the amount of heat required to raise temperature of 1 gram of substance by 1 degree Celsius, meaning that in order to increase the temperature before your thermometer can reach, the energy needs to be absorbed first by the water molecule. The moment it is saturated, then there is a balance, right? Yeah, the balance will be detected by your thermometer, and that's the increment of it. And you can see from here, water, the heat needed in form of joule per gram Kelvin is actually rather high. To show you, it's not easy to increase the temperature of water. You need 4 joule. Look at the ethanol. That's like only 2. Water solid, that's mean ice. Water. How about aluminum? This is for aluminum you use to wrap your barbecue meat because it can easily absorb heat. When it can easily absorb heat, it can quickly cook your barbecue. Yeah. Imagine if you wrap your barbecue with something of high specific heat. For example, you wrap your barbecue with fireproof blanket. Can it cook?
No.
Why not?
It's quite.
It's very thin. It's only 1 mm thin. Still tak boleh lah. That thing specific heat dia sangat tinggi sebenarnya. You need the heat as much as you want to melt the iron. Super high. Then only you can cook your barbecue meat in it. Right. Yeah. So 1 calorie is that much. If you expose it to gold already becoming this temperature. But the same 1 calorie, when you expose it to water, there will be barely any increment in temperature. To show you that this is the very reason why plants use water to transpire, why your body is filled with water, why agriculture you need to water your plant. It's not only because your plants need to dissolve all the nutrient in the soil. It's actually for cooling. Did you know that for 1 kilogram of rice, do you eat rice? I think this year I've only eaten rice once. Itu pun sebab dia beli. Why? I don't eat rice. I do study rice, but I don't eat rice. Aku tak boleh. rice is not my staple food. Okay. Apa benda aku cakap tadi? Lupa dah. 1 kilogram of rice. Do you have any idea how much water is needed to produce 1 kilogram of rice? How much water in the agriculture field? 1,440 liters of water per kilogram of water. Per kilogram of rice. Do you have any idea 1,000 liter tu banyak mana? What's your blood volume in your body? It's about 6 liters. About 6 liters. So it's plants require so much water, not for the photosynthesis so much, not for to absorb the nutrient, but to cool itself. Okay, because metabolic process gets very heated at times. Okay, and Malaysia is very hot. Right. Okay. So, what's this? So this is the electron micrograph of a leaf surface. You got your, what's this? It's a stoma. Breathing out, in and out, oxygen, CO2, H2O in the form of gas. Right. So the transpiration takes place, does it take place at night? Transpiration. Does it happen at night? Transpiration, bukan respiration. Who says transpiration happens at night? Both day and night. Tak ada. So aku seorang je angkat tangan. Okay. Who says it's only happen during the day? Tak angkat tangan juga. So, which one? Day and night. No, seriously, do you think does the transpiration happen during the night time? A little. Hmm. Yes lah. Stomata tu ada je. It's not like at night stomata punch out then go back home. Stomata still there. Ada. You got stomata, you got lenticel, you got hydatodes. All these are holes on the plant body that can allow the water to exit. Right? So transpiration do happen, but at significantly less compared to day. Okay, so at night it's not really for cooling, but to bring nutrient up, pulling. Because remember, water can pull the water before it, so that is needed to ensure that water supply keeps on coming to the leaf, so that the leaf is not wilting; it stays fresh and turgid. Right. And letterhead. Should I go here? Um, I'll just you should know this. You have taken your fundamentals, right? Right. Yeah. Okay. So this is more relevant if you were to live in temperate countries, because in temperate countries you need to worry about the frost. Do you have frost here? Tak ada. Are you sure? Only in winter. Okay, so you started off as a solid ice, then you can see that the temperature increase, then it will start to melt. You see, once it has melted, even though you progressively providing heat as energy, look at the temperature. Does it increase? This is what we call as latent heat. Latent means kind of like a dormant. It keeps on absorbing the energy, but when you measure the temperature, it's not increasing. Because the bonds, remember the bonds earlier? As long as the bonds are absorbing all the energy, then you are not going to see any increment in temperature. So this is why the graph here becomes flat. The moment that has been satisfied, then only you will change in phase from the ice here. You got your liquid, and then the liquid will start to absorb the energy again. Then you got your condensation. Okay, then this go on for some time before your liquid change into the gas form. If you further provide the energy to your gas, what's going to happen? It starts as a solid ice. Now it becomes the water, the liquid. Then you provide the energy again, it becomes the steam, the gas. This steam, this gas, if you further provide the energy, what's going to happen? This axis can go on. What's going to happen to your water vapor?
Gone.
Gone? Gone where? Mari. Mana? Fourth state of matter, plasma. Ke tak belajar? Tak ingat. Now I tell you, ingat. That actually happen in nature. Tu kilat tu, plasma lah tu. It glows because that molecule now, it need to change the state from the gas molecule. It absorbs so much energy in it. That energy gets excited. It needs to go back to the ground state. Masa nak go back to the ground state tu, poof, light splattered about. Then you get your lightning. So lightning is actually plasma. So the fourth state of matter, hari ni kau belajar. Fourth state of matter, plasma. Solid, liquid, gas, plasma. Ada nombor 5? Tak apalah. Sebenarnya nombor 5, tapi itu kena ambil kelas fizik. Right? Okay, so why this is important in agriculture? Why this is important in agriculture? To know this about the latent heat. Why? Kenapa? You see in temperate countries, sometimes the frost can get a bit out of hand or early frost. So when that happens, if the farmers have vegetable growing, they need to protect the vegetable from this frost. Okay, if the frost get the best of the vegetable, it will turn into ice in the cell. That's fine. This ice not smooth, it's jagged, it's sharp. It will start to puncture all the cells in the leaf. And also it can rupture the cells. And when this happens, your vegetable produce is going to turn up mushy. Is that sellable? Can you sell it? Boleh kan? So farmers perlu letak blanket over the crops during the very cold night. You don't experience it here. So boleh tell story sajalah. Then what about the other part of it, the gas, the vapor? If you were to grow plants in Sahara Desert, is that possible?
No.
Do you know that people are growing crops now in the desert? Not because of the oasis. When they understand how you can capture water, you can actually reverse this. You can actually reverse this. So they have like the planting area, and then they start to put the material that naturally cooler than the surrounding temperature. When the desert wind come along, in the wind, there's a water vapor. However, this is not accessible by the plant. However, when the wind hits all this structure, condensation start to take place. Then the water will appear on the surface of the material and then get collected at the bottom, and the farmers can use the water. Nampak tak? Sebab tu ilmu macam ni penting untuk pertanian. You can't, I can understand you can't appreciate this sebab you don't get sandstorm in Serdang. Or do you?
No.
What kind of storm do you get? Lecturer storm, being out of the class. Sebab apa? Emo. Alright? Okay. Right. Ni tak payah. If I tell you masuk exam nanti, so far kan ada seorang je jawab soalan ni betul. Surface tension. So surface tension roughly. Don't worry, tak masuk exam. I just explain. What is the force needed to spread this? Let's say that this is water, right? How much force you need to spread this water molecule using this equipment before it breaks? If you have two droplets of liquid, one you have water, one you have dodol, which one can you need force more to spread for 1 meter before it breaks? The water or the dodol? Why? Because it's more viscous, right? So the surface tension of dodol is a lot higher than water. But if you compare water maybe to something, let's say here, helium, liquid, water, surface tension, 72. Yep, hydrogen. Hydrogen, liquid. Hydrogen can turn into liquid. You only baru move a bit, dah break. So you need a lot less force to thinly spread it. Okay. So this gives the properties of water to form the skin like here. That's why things can float. And also the droplets form when you spray any solution onto the leaf. Okay, so when this happens, the droplet, rather than spreading, it will curl up and form the spherical, not really spherical, the teardrop shape. Okay, so why this is important in agriculture? Have you taken your wheat science? In which science you're going to learn about a concept of adjuvant. Adjuvant is the substance you add into your pesticide, so that when you spray your pesticide, it's not going to form droplets on the leaf, but it's going to spread out. When you allow your pesticide solution to spread out on the leaf, it can be absorbed better into the leaf and do the job. If it stays in the form of droplets, will it do the job correctly? Boleh tak? Dah tak faham. Itulah aku suruh balik bergembira tak nak. Nak ajar dah tak faham. You spray pesticide to your plant. One with adjuvant to cause the water to spread evenly on the leaf. One without adjuvant. When you spray, it becomes like this, droplets. Which spray is going to be more effective, the pesticide? The one forming the droplets or the one without the droplets? Without. Why? Because it increases the surface area. So the pesticide can do the job better rather than it gets clump up the water. It gets clump out, it's fine. Then it drop on the ground and then your cat eats it. Then you were hoping to defoliate your pesticide. Now your cats get defoliated. What was once the British short hair now becomes a Sphinx cat. You know Sphinx cat? Right? Okay. Okay, so this will be the last, okay? Sian korang. Itulah lain kali jangan minta. Okay, cohesion, adhesion. This is the water properties. Water is absorbed by the root, and the root is going to absorb the water, and it needs to reach all the way to the top. And this is like 100 meter up. Does plant have heart to pump the water?
No.
So how come it can pump? 100 meters is very tall.
Is it water pressure?
Water pressure. So a number of things happen here in terms of water properties and also the law of water potential. Baru nak masuk water potential. Adhesion, cohesion. Cohesion, the water on the upper level of the plant pulling the water in the lower region or at the back. So it's like your friend pulling you to move forward. That is one thing. Number two, the water sticking to the wall of the xylem, like this, so that it's not falling due to the gravity. Number three, due to the capillary action, because xylem is very small. Capillary effect. When you have two straws, one 1 cm diameter, one is 0.5 centimeter diameter, you put it both straws in the water container, which straw is going to have a higher? 0.5 due to the capillary action, right? And the fourth one, why the water can travel up? Due to this thing. So that's the unit for water potential. Water potential unit is mega. PA stand for what? Pascal. Okay. The rule is water will move from higher water potential to lower water potential. Pure water, pure H2O, 0. Okay, so what is this actually? When we talk about water potential, it is actually the ability of the water to do job. To do what job? To move according to the concentration gradient. And it follows from the high gradient all the way to the lower gradient. When you have two systems, one, two, three, four, five, six, 1, 2, 3. Both is H2O. Times 3 times 6. Which system has more H2O? 6, right? So we call that this system has more water potential because it is filled with more water molecules. When you bring this thing close together, let's say that we bring together closer, and there's a semi-permeable membrane, water going to travel from which direction? Left to right or right to left? From the left to the right. Yeah, and now eventually they're going to become ni ada berapa? 6, 3, 9. So 1, 2, 3, 4. 1, 2, 3, 4. Tengah-tengah seorang tu. Can you see that? So when you have a high water potential, means that that system is containing more water molecules. As simple as that. Pure water, always 0 water potential reading, 0 megapascal. Can water potential go negative?
No. Yes.
No? No, yes?
No.
No? Kalau no tu, I need to be worried lah. Tengok tu berapa? Okay, please get this correct, okay? Because ada orang dalam Viva tak berhenti jawab soalan ni. I ask a simple ni je. Water potential by default is 0, but it is almost impossible to get 0 water unless you are in a protected lab, pure H2O. The moment a speck of impurities present in water, immediately water potential becomes negative. Yeah, so all water in our system by default negative in reading because there's always impurities in there. Kalau tak ada habuk, there's a bacteria in it. If there's no bacteria, ada virus. Always negative. So when you have two systems which both have negative, the rule is the water will travel from the more negative or less negative? More negative to the less negative, approaching 0. The example is in here. Look at the root here. Water that is present in the root region, the reading is what? Negative 0.2. Water at the top of the atmosphere, at the top of the canopy here, what's the reading? Negative 100. Which one is close to 0? The root region or the atmosphere region? Okay, so when it is close to the 0, it means the water has the higher potential compared to the water that is at the top of the plant now. So the rule is it will travel from the less negative 0.2 megapascal to the region of more negative 100 megapascal. So it will travel that way. Both is negative because both is not pure water. The water got nutrient, got hormones, got so many stuff in it. Right? So it will follow that way. Can the water potential become positive? Macam mana? Boleh tak boleh? Let's leave it to the next lecture for you to find out. Alright? Okay? Alright, okay. That's all for today. Alright. Okay. Okay. Any question? Just any regret? I think yes. Right, okay. So I'll see you on Monday, okay? Okay. What you need to submit next week?
Oh, NGR. Oh, ada the calculation.
Yeah. Yeah. I might suddenly ask, nak tengok? Jangan kata aku tak ada. Show to me your work. You need to print ke apa, make it happen. Okay. Alright, that's all for today. See you.